In this video, we will discuss about how this O-glycan formation occurs in the cells and what are the changes in the O-glycans occurs in the tumor cells compared to the normal healthy cells. So this is the beginning of the O-glycosylation. That is, this GalNAc will binds to the serine and threonine amino acid of the translated proteins, and this glycan structure is known as TN antigen, and thereby with the help of this core 1 GALT1, this addition of this galactose at beta 3 position occurs, and this structures is known as this gal beta 3 galnac alpha structures, and they are commonly known as T antigen core 1. So this is also found in the normal cells. And however, in the case of tumor cells, what happens? There is a upregulation. There is a transformation from this gal beta 3 galnac alpha to with the help of this ST3 galnac enzyme. This addition of sialic acid occurs to this gal beta 3 galnac alpha, and these structures, that is sialic acid alpha 6 and gal beta 3 galnac alpha, is more evidently found in the case of tumors, and this is commonly known as silyl T antigen. And the other structures that are also known as silyl T antigens. However, in this case, the bond is slightly different. Instead of this sialic alpha 6 bonded to the galnac, it bonds to this gal beta 3, and this is done with the help of this enzyme ST3-GAL1. So there is upregulation of this ST3-GAL1 that leads to the more structure formation of this silyl alpha 3 gal beta 3 galnac alpha. And however, there is a downregulation of this structure, that is galnac beta 6 to galnac alpha, gal beta 3 galnac alpha, and this is because there is a down regulation of these C2G and T1 enzymes, and this is the reason why this further down regulation occurs, that is towards this core 2 extension. However, moreover, this core 3 galnac T enzymes down regulation has also been observed in the case of tumor cells, and this leads to the further down regulation of this kind of structures that are not found, that is core 3 structures, and core 4 structures are less found in the case of tumor cells compared to the healthy cells. Now let us quickly cover how this formation of this Lewis antigen occurs. So in this case, this Lewis antigen formation begins the backbone of this, or the initial point of this Lewis antigen formation is this glycanac beta, and from this glycanac beta, they with the help of this enzyme beta 3 gal T and beta 4 gal 1, this lead to the formation of this gal beta 3 glycanac beta and gal beta 4 glycanac beta respectively. After that, since there is upregulation of this ST3 gal, so it will lead to the addition of this sialic acid at alpha 2 3 position, and this leads to this structures formation, that is sialic acid alpha 3 gal beta 3 glycanac beta and sialic acid alpha 3 gal beta 4 glycanac beta, and thereafter there is a addition of this fucose at alpha 4 position with the help of this alpha 4 FUT enzyme, and this leads to this final structure formation of this sialic alpha 3 gal beta 3 glycanac beta FUT 4, and this FUT enzyme is usually FUT 3 enzyme, and whereas in the case of this Lewis S structures formation, this alpha FUT 3 will use an in this case the enzyme that has commonly been called as this FUT 3 and FUT 7 enzymes that results in the formation of this silyl level X structures, that is sialic acid alpha 2 3 gal beta 4 glycanac beta, and this is bonded to this fucose alpha 3 monosaccharides, and this structure is commonly known as silyl Lewis S structures, as it is as fucose is bonded to this alpha 3 bond. However, when this fucose is bonded to this alpha 4 bond, then this structure is known as silyl Lewis A structures. And as I have already mentioned, these silyl Lewis A structures is uh, promoted by or is formed by the presence of enzyme FUD3, whereas level S structures is formed by the or is formed by the enzyme FUD3 and FUD7, and this is formed by FUD4. Lewis A is formed by FUD4, and Lewis X A structure is formed by FUD3 and FUD7. So I hope you have understood how this O-glycans formation occurs in the cells and how they are differ in the case of tumor cells. If you need to understand this O-glycosylation in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.